Everyone, Mr. Jennings here, and today we're going to talk about population evolution. Now, population evolution is the next logical step in evolution discussion because first we already talked about what is natural selection, which is the main force of evolution. We talked about the other forces of evolution, such as sexual selection and artificial selection, as well as bottlenecking, genetic drift, and gene flow. Next, we talked about the evidence to support evolution. We talked about things like embryology, biogeography, various anatomy structures, and the fossil record. Next, though, we're going to talk about what happens on evolution on a large scale. See, a single individual cannot evolve. Evolution is a population force. It's how it works. Typically, you need a big group of individuals because of one thing. See, for evolution to happen, you need variation. Now, variation within an individual is one thing, but what we're talking about is variation within the gene pool. See, variation within the gene pool is any change in the genetic code that makes you unique. You need variation. Without this, you will not have evolution. Without variation, you can't have different forms, different traits for selection to take place. You can't select from only one option. You need multiple options. Now, the main way to get variation are mutations and sexual reproduction. There's a typo on the screen. But mutations and sexual reproduction are the primary ways to get variation within the population. Because mutations are any kind of change in genetic code, and sexual reproduction is a recombination of two different organisms' genes. And sexual reproduction is also the added benefit of every successive offspring outside of like twin situations, identical twins. You're going to get a situation where every offspring is going to be genetically different from the previous offspring. So it's pretty great. Problem with mutations, though, mutations are rare, especially in starting into eukaryotic organisms. Mutations that are meaningful are exceptionally rare. That's a better way of saying it. Because one, your DNA has redundancy. All right, so let's talk about what we're doing. Let me show you an example here. So in DNA, you had, AUG. That's a genome. That's a codon. Well, that third letter, in most cases, doesn't matter. It does here, but let's say we're talking like UUC, UUA, UUG, and UUU. All four of these, the third letter rarely matters. This letter here is often replaced and just replaced with the letter N in most cases. So mutations that cause variation need to take place in the second or first base of the codon. So that's rare. Two, they also have to happen in the germ cells, because if these mutations don't occur in the germ cells, they can't be passed on. It has to be able to happen in cells that begin up producing sperm and egg cells. So you have to have a mutation. Not only do you have to have a mutation, the mutation needs to happen in a cell that can be inherited. That's really hard. So variations take a while to start showing up through mutation, but sexual reproduction creates a lot of variation. Now, in modern genetics terms, evolution is defined as a change in the relative frequency of alleles in a population. So change the frequency of genes in the gene pool, you have evolution. That's pretty straightforward. If that genome has changed, that gene pool has changed just slightly, more likely you have evolution. And there's ways to calculate that, but we're not going to get to that at this course. But we're going to talk about how certain genes are affected. So there's single genes and polygenic genes, traits. So single gene traits are traits that are produced by one gene. Simple stuff like, T equals, and this example here is widow's peak. Widow's peak, lowercase t is no peak. And you have, two phenotypes. Big T and big T, gives you the widow's peak. And little t gives you no widow's peak. That's simple single gene traits. We call these Mendelian traits that are only controlled by one gene. At most, you see 3 phenotypes because there's a heterozygous phenotype, either in co-dominant or complete dominance. Most traits, though, are going to be controlled by 2 or more genes, and they're called polygenic traits. And these are the traits that look like this. Because they have 2 different combinations or more, the possible phenotypes change. Typically, in what we call a dihybrid crosses we see here. We see situations where you have four possible phenotypes under normal situations under Mendelian forms, but if they're polygenic traits, you can get quite a bit more. So it can vary. Now, keep that in mind though. So when we look at certain things like natural selection on a single trait, we have a classic example of the peppered moth. So this is what these are. They're the peppered moth. They're found in England. Now, in England was undergoing the Industrial Revolution. Industrial Revolution produced, where are the mouse? So you have a bunch of factories. They're battling out coal smoke. They got over to the trees. And the trees were going from white trees to black trees. So what happened? There was these little moths that stayed on these trees. The peppered moth. Now the peppered moth, under its natural form, is what we see here. You can't see it's in this little circle. Naturally blends in with the tree. Pretty awesome. Now there's a mutant. This here is our mutant. This mutant is known as a melanistic individual. It's like the opposite of albinistic. Melanistic means you're fully pigmented. Everything's pigmented. And in this situation, if you're a predator, you're going to go for the first thing you see. The melanistic moth is easy to see. It's going to be eaten. Now, is that melanistic moth always going to die? Probably not. It's going to die more often than the white moth. So the white moth here, in this case, the normal pepper moth, is pretty common. However, in the Industrial Revolution, remember, when the factory smoke started polluting the trees went from white to black because of all that smoke. So when all this started filling in, the peppered moth actually started to blend in really nicely. Let's see if I can change it to a black pen. So when all this started filling in, the peppered moth, that was the melanistic individual, started blending in really well. Meanwhile, the white form, the natural wild type, was starting to stand out more. So predators were starting to lean towards from the black moth to the white moth. Because predators really don't have a preference besides which is the easiest to catch. If it takes the least amount of work, they're going to do it. Sticking out like a sore thumb, least amount of work. So when we saw this, so this changed a frequency from being, let's say, before the Industrial Revolution, you had, let's say, in a population of 100, you had like 75% white. That's way too high. And let's say you had, 25% black. This is pre-Industrial Revolution. After that Industrial Revolution took place, they flipped. So the white, it flipped like this. 75%, 25%. Because the white moths now were sticking out and the black moths weren't. Then you go, Mr. Jennings, was that always true? And the answer is yes, it was always true. Because what's interesting is, once the Industrial Revolution environmental regulations started taking place, the trees went back to white as new growth was starting to happen. And we saw the flip happen again. So the trees went back to white, and all of a sudden the black moth populations decreased and the white moth populations increased. This is an example of what we call a single gene evolution. Because the genes changed slightly, the gene pool changed and we had new individuals. So that's one example. Just how that goes is a classic example. Now, when you have polygenic traits, things get complicated. See, polygenic traits don't fall on a simple bar graph like we see here. This tends to be, these are your monogenic single traits. Let's say, polygenic tends to look like this. You can put it in a bar graph, but it tends to be a little bit more diverse because you're going to see more phenotypes. And usually instead of drawing the bars, they'll just draw a curve because it's a little easier to see the trends. So keep that in mind. Everything we're going to talk about from here, these types of selection, are all dealing with curves. And here we see this. So the yellow curve represents the original. So let's say you are talking about individuals that are tall and short. this side being short, and this side's being tall. What this graph is saying is very few plants are really short, and very few plants are really tall. Most plants in this original population tend to be medium size. And that tends to be the case. This is what we call a normal distribution, or a bell curve. Typically, more individuals will favor the average than they will favor the extremes. Now, let's say something happens that the tall plants are being favored for. I don't know, lowland grazers, insects or something. So that's causing a selective pressure. So this downward arrow is what we call a selective pressure. Now, selective pressure is like it sounds like. It's some source that is selecting, either selecting for or against a certain trait. In this situation, it could be a low-lying insect pest that is eating the shorter plants. So they're going to tend to favor the taller plants. So it's better to be taller. So the population is going to shift towards the tall side, where the average all of a sudden becomes taller than the original average. So that's the shift. Now this can happen in both directions as well. This is called directional selection because it shifts the curve in one direction. Now next we have stabilizing selection. Again, this here is the original population. We don't see the new one as much, but we see the curve. And this is called stabilizing. See, stabilizing is a pressure on the extremes. So let's say, this is the number of individuals with the phenotype. Again, we'll still keep our tall and short going because this example still works there. Let's say being too tall, you're all of a sudden more apt to getting eaten by large predators, and too short, you're being more apt to being eaten by small predators. So you are better being in the middle, the Goldilocks zone. So if you want to think of this anyway, it tends to be the Goldilocks. It works perfectly fine. This is, the medium is better. And we see this a lot in nature. This tends to stabilize, is what it says. It keeps the population being average. An example of this in the real world, in humans, is sickle cell anemia. Because in humans, if you are, and sickle cell disease is represented by the HbA, HbS notation. Break down that because you may not remember. HbA means normal, and HbS lowercase is sickle, and HbS is lowercase. Is a homozygous recessive trait. Now, what happens is if you are HbA, HbA, you're normal, but malaria prone. You can catch malaria in certain parts of the world. Specifically, East Africa is where this study was found, but we found similar results elsewhere. If you were HbS, say you're HbA, HbS, you tend to be, you're still normal under normal conditions, as long as you weren't going hypoxic, means low oxygen, but malaria resistant. So you could handle malaria. You actually couldn't catch it at all, hardly. Low infection rate. But let's say you were an individual that was HbS, you're going to be malaria resistant, but you're going to have a more serious disease known as sickle cell. Sickle cell is more dangerous. It can cause more havoc to your body. It's more likely you're guaranteed to have it. Versus malaria, you're not always guaranteed to get it. And there's medication for malaria. Sickle cell, there's the medication's there, but it's more of a treatment, not a cure. Alright, so keep that in mind. Now, one thing I want to say this here is, these are single gene traits, but this same premise. Heterozygous is favored, because being a combination of both is great compared to being homozygous for either trait. Now, disruptive selection is this one right here. Couldn't find a good picture of it with all three listed. Disruptive selection is the average is selected against. Some reason it's better to be the extremes. Being a middle ground or, average whatever phenotype tends to be not great. It's better to be hot or cold than it is to be lukewarm in this situation. So it's very disruptive. Yeah. Both disruptive and directional can create what we call speciation events, and stabilizing tends to keep the pattern true. Oh, too far. Now, change in genetic frequencies, those ones are just listed here. They can also be compounded with things like genetic drift and founder effect, which we already talked about. Founder effect is when a new population moves in, tends to colonize, and bottleneck is a disaster. Either one of these can cause genetic frequency changes, but these are a big thing about these two is genetic drift in general is what we call a random event. It doesn't have any kind of preference for either certain either any phenotype usually. So this is a random shuffling of the genetic frequencies. The previous ones we were talking about were selective. Disruptive, stabilizing, and directional are selection. They favor for or against a trait, or phenotype. So that is the difference between these two. Either way, we can use what we call Hardy-Weinberg equilibrium to discuss this. Hardy-Weinberg equilibrium is complicated. If we weren't to get into the math, I'm just going to show you the equation so you can see it. This here is this is what we call homozygous dominant. This is your hetero. And this is your homozygous recessive. The way we get this equation is by just basically doing a binomial. We're going to spare. Anyway, what you can do is look at the numbers compared to the original population to the new population, and if there's a statistical difference, you have evolution. That's how that works. But that's beyond the scope of the course. State of Tennessee just says I can mention it to you. I could talk about it if we want to. I don't see the purpose, at least at this level, because it's kind of a challenging thing for most students. So we're not going to talk about it too much. But we will talk about it in this regard. So that's fine. All right, let's kind of summarize here. So natural selection and evolution in general are major changes to the gene pool frequency. Now, today we talked about how the gene pool can change. So you can have single genetic traits where one trait's selected for it against, and that can cause some shifts. You can also have where it's polygenic. Now, the polygenic ones also can work for like the ones with 3 phenotypes. Basically, you need to have more than 2 phenotypes to have a polygenic curves. But you have any situation where you have directional, disruptive, or stabilizing selection. Those forms of selection change the gene pool. And when you change the gene pool so much to a statistical level, you get evolution. These other ones that we see on the screen here, genetic drift, founder effect, and bottleneck effect, but also migration, gene flow, all of them can cause changes to that Hardy-Weinberg equation that you see right here. Be aware of that. Other than that, though, that's all I got to say today and this week. This week is a little bit longer video. We'll have something else to go along with it. But take care. Have a great one.